Meiosis 1. The first step of meiosis 1 is prophase 1. This is where chromosomes become visible, crossing over occurs, and the nucleus disappears. The meiotic spindle forms, and the nuclear envelope also disappears.
Chromosomes occur in meiosis between phase 1 and metaphase 1 and is the process where homologous chromosomes pair up with each other and exchange different segments of their genetic material to form recombinant chromosomes.
The third step is anaphase 1, which begins when the two chromosomes of each tetrad separate and start moving towards opposite poles of the cell. As a result of the action of the spindle, notice that the anaphase 1, the sister chromatids, remained attached as their centromeres and move together towards the pole. The last step is telophase 1. At each pole during this stage, there is a complete haploid set of chromosomes, but each of these chromosomes still has two sister chromatids. A cleavage furrow appears, and by the end of this stage, the parent cell has divided into two daughter cells. This separation of the cytoplasm is called cytokinesis.
But chromosomes do not replicate again because they already replicated. Prophase 2 is characterized by loss of the nuclear membrane and nucleus, spindle information, and the appearance of rod-shaped chromosomes. Metaphase 2, chromosomes line up at the equator of the spindle. Each chromosome consists of two sister chromatids joined together at the centromere, which is attached to a spindle fiber. Anaphase 2 begins with the separation of the centromeres. The sister chromatids move towards opposite poles of the spindle, now called daughter chromosomes. And in telophase 2, they form new nucleus, and cytokinesis divides the cell to form two haploid daughter cells. Because each cell entering mitosis 2 forms two daughter cells, a total of 4 haploid cells are produced from the original diploid parent cell during meiosis 1. Thus, meiotic cell division may be summarized as one cell, and then it replicates into 2, which then creates 4 daughter cells.
Process where cells have a distinct nucleus, and in the G2 stage, 2 pairs of centrioles. The chromosomes are uncoiled and are visible as chromatin granules. In prophase, during this phase, the nuclear membrane and nucleus disappear as rod-shaped structures, and the replicated chromosomes coil tightly. Then each pair of centrioles migrates to opposite ends of the cell. The fourth step is that microtubules extend between the parts of centrioles. Astral rays constitute an aster, and each end. In late prophase, spindle fibers from opposite pores attach to the centromeres of the sister chromatins. In metaphase, this is characterized by the chromosomes living up at the
Two key differences between meiosis and mitosis is that meiosis has 2 round genetic separations and circular divisions, while mitosis only has 1 each. In mitosis, the daughter cells are identical to the parents as well as to each other.